Good afternoon, students. Today we are going to discussing lipid metabolism, certain disorders, also amino acid metabolism. In amino acid metabolism, general reactions, then urea cycle, catabolism of phenyl alanine and triosine, and phenyl ketonuria. So first one, atherosclerosis. Atherosclerosis means it is a disease condition in which the plaque is deposited or built in our arteries. Plaque means it's a fat or a cholesterol, and this plaque will result in hardens our arteries and they will narrow our arteries, and this may result in heart attack, stroke, even death also. See, plaques are mainly of two types. One is slow growing plaques, and this will not interrupt our blood flow, but over the time they result in chest pain. Then only we realize that we having block or clot. Another type of plaque means they will get suddenly rupture and results in stroke, chest pain, etc. So plaques of atherosclerosis, there are three types, that is coronary artery diseases, cerebrovascular diseases, and peripheral artery diseases. So coronary artery diseases means the plaques will get ruptured and the clot will get formed and they result in chest pain, etc. Cerebrovascular disease, this is happening in our brain. Then peripheral artery diseases means happening in our leg. Same process, the plaques get ruptured in the artery of leg. Then what are the causes? Smoking, high cholesterol, high blood pressure, diabetes, obesity, stress, not having proper food, excess alcohol, not exercising properly, etc. Treatment, lifestyle changes, that means have a proper food, proper exercises, etc. Medication means drugs that is prescribed by doctor based on our cholesterol level, blood pressure level, etc. Next is angiography and stenting. That means angiography means angiogram, that is a tube is inside in our arteries. So this may result in smooth blood flow. Then stenting. Stenting means a stent is inserted in the blocked artery. Bypass surgery. Bypass surgery means a healthy blood was taken from our leg or chest, etc. And this is bypassed with the segment. Next is fatty liver. Fatty liver means, usually the liver means they are the storage organ, that is adipose tissue. So the concentration of lipids in our liver is about 5%. So in normal liver, the Kupffer cell, they'll produces droplets of the lipid. But in fatty liver, the droplets contain triacylglycerols. So this results in fatty liver. Also, they are associated with fibrotic changes than cirrhosis. Fibrotic means the thickening of tissues, etc. Cirrhosis means it is a chronic disease condition of the liver by inflammation or thickening of tissues, degeneration of cells, etc. So mainly these fatty livers are due to two reasons. One is increased synthesis of triacylglycerols. Another one is impairment in lipoprotein synthesis. Increased triacylglycerol synthesis. So from adipose tissue, the free fatty acids are mobilized and they are moved to liver. So while these fatty acids are overproduced and they will get accumulated in liver and results in fatty liver. Next is impaired synthesis of lipoproteins. That is, for the proper synthesis of lipoproteins, we require phospholipids, apoprotein B, etc. So, when lipoprotein synthesis is get impaired because of the defect in the phospholipid synthesis and blockage in the apoprotein formation, etc. So, these lipoproteins are associated with certain factors, that is endocrine factors, then lipotropic factors, etc. And so these factors, they deposit in the fat in the liver. So due to this, fatty liver will causes. Then choline deficiency and fatty liver. Same thing is explained. There is phospholipid, apoprotein, etc. Then fatty acid oxidation will also get impaired here. Next is obesity. Obesity means there is excessive body weight. This may result in negative impact on our health. So usually our weight should be based on BMI. BMI means our body weight is divided by square of our height. So that is BMS. So always we should follow BMS. Then this obesity is linked with other diseases like cardiovascular disease, diabetes, then sleep apnea, then cancer, osteoarthritis, then depression, etc. What are the causes? Improper food, improper exercise, also genetics, then medical reasons, then psychiatric illness. Management, we have to follow proper diet and proper exercise. Then bariatric surgery, it is very effective surgery in the case of obesity. Okay, next one is amino acid metabolism. So in that, general reactions of amino acid metabolism, that is transamination. Transamination means here amino group is transferred to keto acid and they form alpha keto acid. This is called transamination. So this is the example. For that we are using the enzyme PLP, that is pyridoxal phosphate. Amino transfers also we are using. That is, from amino acid 1, amino group is transferred to form alpha keto acid 1 using PLP. Then from amino acid 2, it is catalyzed by transaminase to form alpha keto acid. So, what are the features of the transamination? Pyridoxal phosphate, it is the coenzyme. They helps in transamination. Then, for each pair of amino and keto acids, there will be a specific transaminase. For example, for aspartate, we have aspartate transaminase, and for alanine, we have alanine transaminase. See, while these amino groups are transferred, there is no liberation of ammonia, and they are reversible. And transaminase is very important for the redistribution of amino acids and non-essential amino acids, and they result in energy generation. Then amino acid undergo transamination and concentrate nitrogen glutamate. Then amino acid like lysine, threonine, proline and hydroxyproline, they will not undergo transamination. Then transamination is not restricted to alpha amino groups. Then serum transaminase, they are very important for the diagnostic and prognostic properties. Then mechanism of transamination. So here amino group is transferred from amino acid using the coenzyme pyridoxal phosphate, and they'll form pyridoxine phosphate, a pyridoxamine phosphate. And from this enzyme, from this compound, again the amino acid is transferred to form 2 keto acid and to form amino acid. So this is the mechanism. Next is deamination. Deamination is nothing. It is removal of amino group. So there are two types, oxidative and non-oxidative. In oxidative, role of glutamate dehydrogenase. So glutamate dehydrogenase. Glutamate means this is the collection center of amino groups. So glutamate rapidly undergo deamination, and this is catalyzed by glutamate dehydrogenase. In this reaction, the ammonia is liberated. So here we are utilizing NADP or NAD positive as a coenzyme. So finally this glutamate is converted into alpha ketoglutarate. Also an intermediate is released, that is alpha aminoglutarate. Next is non-oxidative deamination. In that first one, amino acid dehydrase. So here amino acids like serine, threonine, homocysteine, they are hydroxy amino acids. So they are catalyzed by PLP dehydratase and they result in alpha keto acids. Next is amino acid desulfurase, that is cysteine. They will undergo reaction with desulfurhydratase, and here the ammonia is liberated to form pyruvate keto acids. Next is deamination of histidine. That is, histidine is react with histidase to form urocanate. Here also ammonia is liberated. Next one is decarboxylation. Decarboxylation means it is a chemical reaction that removes the carboxyl group or releases carbon dioxide. So you can see, so from this amino acid we are removing the carboxyl group to form amine. So these are the examples for the decarboxylation, that is phenol aniline to phenyl ethylamine, triosine to trimine, etc. So one of the examples here you can see triosine decarboxylase. Here the carboxyl group is eliminated and form trimine. So likewise histidine, the group is removed to form histamine. Next, metabolism of ammonia. See, ammonia is liberated during the metabolism of amino acids or other nitrogenous compounds. So at pH, physiological pH, ammonia exists as ammonia. In that first one, the formation of ammonia. So here the ammonia is formed from amino acids like reactions like transamination, deamination, etc. How the ammonia is formed with the help of intestinal bacteria on urea. Next is transport and storage of ammonia. So here the constant production of ammonia from various tissues. Its concentration will be very low, that is 10 to 20 mg per dl. this is mainly because of ammonia transport and their utilization for the urea synthesis. That is why it is very low. So this ammonia is transported through various tissues and other liver, and they results in the formation of glutamine or any other amino acids, alanine, etc. So alanine means it is a very important amino acid for this ammonia transportation. So glutamine can be deaminated by hydrolysis to release ammonia. Next is disposal of ammonia. How the ammonia is disposed, that is example ammonia tele, that means aquatic animals dispose ammonia in the water. Then uricotelic, that means through uric acid they are converted, birds, reptiles, etc. The ureotelic, that is in mammals, can convert ammonia to urea. So these urea are very non-toxic and soluble, so they are easily extracted. Then toxicity of ammonia. Toxicity of ammonia means the blood ammonia concentration is very harmful to our brain. So these are get accumulated in our body. This may result in slurring of our speech, blurring of our vision, etc. Finally, death may also occur. So these reactions can be catalyzed by the glutamate dehydrogenase. So this ammonia accumulation, they are associated with more glutamate formation. So more glutamate means more utilization of alpha ketoglutarate. So alpha ketoglutarate is a very important intermediate in citric acid cycle. So finally the ATP is produced by the brain, and these toxic effects are impaired in the ATP production. So this is the metabolism of ammonia. Then urea cycle. So urea cycle means it is an end product of protein metabolism. They are synthesized in our liver and transported to kidneys for excretion. So this urea cycle is found by the Hans Krebs and Kurt Henseleit, that's in 1932. So urea cycle, we can call it as Krebs-Henseleit cycle. So urea having two amino groups, that is one is derived from ammonia and other from aspartate. And the carbon atom is supplied by the carbon dioxide. Actually, this is a 5-step cyclic process with 5 different enzymes. So first 2 enzymes is present in mitochondria and others are in cytosol. So let's see the cycle. So in mitochondria you can see 2 enzymes and other will be in the cytosol. So let's start with mitochondria. So here using carbamyl phosphate synthase 1, that is the first enzyme. They are forming carbamyl phosphate. So here they are releasing phosphate and you can see ATP is converted into ADP plus PI. Then carbamyl phosphatase using orthonine transcarbamylase. They are converted into L-citrulline. And this L-citrullinase goes to cytosol. So using arginosuccinate synthase, we will get arginosuccinate. In between, ATP is converted into AMP plus PPI. Also aspartate is released. So arginosuccinate will react with arginosuccinate lysis and fumarate to form L-arginine, and L-arginine will react with arginase 1 to form orthonine. Here the urea is formed, the water released to form urea, and this orthonine will move to mitochondria to form. So using orthanotrans carbamase L-citrullosamine, this process is continued. So urea cycle means it is irreversible, and you can see that they consume 4 ATPs. You can see 1 ATP here, 1 ATP here, 4 ATPs, and 2 ATPs utilized by this carbamyl phosphate synthase, and 1 ATP for this AMP and PPI, and another one is for the formation of arginosuccinate. They are equal to 2 ATP, that's why 4 ATP. So this is the reaction of the urea cycle. Then normal blood urea concentration is, that is 10 to 40 mg per dl, and 50 to 30 gram of urea is excreted in the urine per day. So there are many tests for this evaluating this urea. There is evaluation of kidney function, Urey's method, or diacetyl monoxamine, etc. So elevation in the blood urea may be broadly classified into 3 categories. One is pre-renal. Pre-renal means what all happening? That is, protein breakdown will be more, and this will lead to negative nitrogen balance, and they result in fever, diabetic coma, thyroxotoxicose, etc. Next is renal. In renal condition, acute glomerulonephritis is formed. It is related to kidney. Then nephrocrosis and chronic nephritis, polycystic kidney. So these are the impairment happening in the kidneys. The next is post-renal. Post-renal means the obstruction in the urinary tract, or reabsorption of urea will be more from the renal tubules. Then uremia. Uremia means more urea level in our blood due to renal failure. Then astosemia. Astosemia means the blood urea or nitrogen metabolites will be more. They may or may not associated with renal diseases. Then catabolism of phenylalanine and triosine. So phenylalanine, triosine, so they are aromatic amino acid. Phenylalanine is essential amino acid, triosine is non-essential. The function of phenylalanine means that is only to convert into triosine. So let's go through a cycle. So the phenylalanine, using phenylalanine hydrolase, they are getting into triosine. Then this triosine will react with triosine transaminase. In between, alpha ketoglutarate is converted into glutarate and form beta hydroxyphenylpyruvate. And this beta hydroxyphenylpyruvate dioxygenase converted into homogenate In between, ascorbate is converting into dehydroascorbate plus water and carbon dioxide is generated. Then this homogenizes will undergo oxidation with homogenized oxidize, and they results in 4-mallyl acetoacetate. And uh, this is react with, that is isomerize, that is mallyl acetoacetate isomerize to form 4-fumaryl acetoacetate. So here they undergo hydration using hydrolase enzyme to form fumarate and acetoacetate. Acetoacetate is converted into fat. This fumarate will go to citric acid cycle and glucose, etc. Then disorders. In that we are discussing only phenylketonuria. Phenylketonuria. That means it is mainly due to deficiency of phenylalanine hydrolase or gene also. Then also in the defect in dihydrobiopterin reductase. Phenylalanine metabolism in PKU. So phenylketonuria, they causes accumulation of phenylalanine in tissues, blood, etc. And they results in the more excretion in the urine. So this will disturb our routine metabolism. So this phenylalanine is diverted to many other pathways, resulting in excessive production of phenylpyruvate, phenylacetate, phenylactate and phenylglutamine. Then biochemical manifestations. What are the happening in CNS? Mental retardation, failure to walk and talk, failure of growth, seizures and tremor. Then impairs the transport and metabolism of other aromatic amino acids. Synthesis of serotonin is insufficient, defect in myelin formation. So this is happening in CNS. Next, pigmentation. Melanin, it is a very important pigment. They are synthesized from tyrosine by tyrosinase. So they will inhibit tyrosinase and they will disturb the melanin formation. Then what are the causes of this? Light skin color, fair hair, blue eyes, etc. Then diagnosis. How we are diagnosis? Screening newborn babies. Increased for checking the plasma levels of phenylalanine. It should be between 20 to 65 mg. Normal is maybe 1 to 2 mg per dl. It is usually carried out by the Gujarat test. So this test is performed after the baby is fed with breast milk for the couple of days. Then phenylpyruvate in urine also can be detected by ferric chloride test. So next is treatment. Dietary food. That means food with more phenylalanine we have to take. Then early diagnosis. Then restriction to protein diet. Administration of 5-hydroxy tryptophan and dopa. So that's about for today. Thank you.